I know I talked about this. Can I get everybody's attention, please? I know I've already talked about the first couple slides, just the idea that we typically learn the most from problems, mistakes, things like that. And then I know I taught you guys a teeny bit about the regulatory proteins. So besides genes that make certain proteins that are needed, there's also specific proteins that are needed, like enzymes and things that turn on other genes. That will make more sense after we start our transformation lab, which we're going to start prepping. I don't know if we'll get to, I think we'll get to some of it today, but I'm not sure. And then, did I talk about cytoplasmic inheritance with this class? Okay, so this is pretty interesting stuff. So once again, please remember that Chapter 14 you're not being assessed on. I just want to explain it to you pretty quickly, and maybe you could kind of recognize it when you hear it, when you're on to another science class, or if it has something to do with your family, some pattern of inheritance in your family, and things like that. Okay, so cytoplasmic inheritance is pretty cool. It's talking about how you inherit all the stuff that's in the cell besides the chromosomes. So, for example, what are these orange things called? The mitochondria. So do you inherit it from your mom or from your dad? Well, so think back during meiosis, and we talked about sperm formation and egg formation. And for meiosis, you've been taught that you get 4 cells, 4 daughter cells. But for eggs, how many eggs do you get for meiosis? 1. We want one good one, right? So you get this little polar body spreading off to get rid of the extra chromosomes. And that's also so that it's an uneven division where you get a nice big egg after 2 divisions. And the polar bodies are just recycled and reused. So that big egg is holding all the cytoplasm. So if your father has a mitochondrial disorder, can you inherit it from him? No. So any mitochondrial disorders then are inherited from the mother's side because of the large eggs. So that's cytoplasmic inheritance. And in plants like cottonwoods, where there is genders, separate genders, you also, they would inherit their chloroplasts from the mother, from the female tree. So do you guys remember the name of these charts? You guys should know the name of these charts. These charts might have been on the science proficiency test Oh, they were. Okay. So what's the name of it? Read it down here. What's the name of them? Pedigrees. So again, the box is at the top here. That's the male, is the square. The trait is represented with the blue. So you could see how the mom, they had a bunch of babies here, and everybody got this mitochondrial trait. Okay, but notice how her sons are dead ends, right? Her son over here married somebody, had 3 children, but they inherited from their mother, not from their father. And here, if the father has it, once again, you see the whole tree there, or that pedigree of being different. Okay, genomic imprinting is pretty interesting. It has to do with other genes on chromosomes. And once again, it's important. Did you inherit that chromosome from your mother or from your father? It does make a difference, okay? So let's listen to this animation.
Normal growth in mice depends on a growth hormone called insulin-like growth factor 2, or LGF2. The reason that the parental origin of each allele matters is a normal process known as genomic imprinting. Heterozygous mice with only 1 copy of the dominant normal allele of the LGF2 gene grow to normal size, parental generation. However, in female mice, imprinting inactivates the LGF2 gene during meiosis. Only the offspring that inherit the normal allele from their father can grow to normal size. Heterozygous offspring that inherit the imprinted normal allele from their mother are dwarfs.
Okay, so for genomic imprinting, what we have figured out is some genes are beneficial if you're a male. Some other genes are more beneficial if you're a female. So that's why it matters. So then they have found examples here where the females have chemically turned off some genes that were good for males. So when they inherited that gene from their father, then there's these chemical reactions that could turn that gene off so that the females are expressing that gene. We see this a lot with the sexual characteristics of attracting mates. So for some organisms, not all, but for some organisms, especially some mammals, but again, not all, it might be better for the male to be larger and the female to be smaller. One fact about being a smaller organism is you need less resources. It takes more energy to be larger. Also, you can't hide as well, right? You can't camouflage as well. And in mammals, the females are often taking care of they're taking care of their children, at least early age, because of mammary glands, it's required, right, to get milk from the mom. So there's these genes are in conflict with each other. If you were a male, you'd want them. If you were a female, you don't want them. And it could be vice versa also. So kind of just some cool stuff here on then the chemistry of turning different genes on and off. And sometimes they can't be turned off or on in this case, and they're just stuck with the genes that they'd rather not have. Okay, epistasis is something you should be familiar with. You saw an example of epistasis when you guys did your baby simulations. So look what it says here. It's when one gene cannot exert its phenotypic effect unless a second gene is also expressed. So do you guys remember what trait it was in humans for our baby simulation? There was a trait that couldn't happen unless you had a previous trait. So there were hair ones and eye color ones and even skin color ones that were a combination. That was multi-genetic, but there was another one that you just couldn't have this trait unless you had a certain trait.
Chin.
Yeah, that chin. You had to have a pretty big chin to get those secondary traits on that end of the chin part. So that's called epistasis. Now something that is also epistasis, but doesn't have to do with structure, it's back to that chemistry, is what if you have a gene that has to make an enzyme to get a certain product made? So you could see here A, this is a reactant, needs to turn into the B products or product. They're just simplifying here. And to get this reaction done, this gene makes this enzyme, this gene makes that enzyme, and we need C. So if something goes wrong with either one of those genes, we're not going to get the product made, the protein made that we need. So that's also another example of epistasis. Okay, this one I think is the most interesting, and I'm not sure how it works. I need to do some more research on it to understand it. But what it is certain genetic disorders get worse as they go to the next generation and then the next generation. And for some of these disorders, originally the disorder didn't show symptoms till later on in life. So this is called late onset disorders, meaning then that these people have already had children and passed on their genes. So natural selection couldn't get rid of the bad genes. The people didn't start getting symptoms till they already had children. Are you guys following me on this? For most disorders, for most disorders that are bad, genetic disorders, the people can't reproduce. They either can't physically or chemically reproduce, like maybe they're not making gametes correctly, or are they just, unable to? Maybe they have growth problems, behavior problems, and anyway, there's all these different reasons that prevent that negative gene, that bad gene from getting passed on, right? Maybe they just died too early, right? And they didn't pass on this gene. That's natural selection, right, you guys? Survival of the fittest. So these disorders that come after reproductive age are not being controlled by natural selection. You guys follow me on that? So what's so interesting to me is, I'm not sure the chemistry on how this happens, but check it out here with this pedigree. Martha got Huntington's disease at she's had the genes her whole life. She started showing the symptoms at age 55. Now, did anybody do Huntington's disease in here? A couple of you, okay? So then, and you might not have realized this because I didn't have you guys do a major research thing, but so Martha had a son that got it. He showed symptoms at 50. Then he had, he had a son, some twins, and then a daughter. And you could see then his son then showed symptoms at 37. Andrew had a daughter that showed symptoms at 26. And you can look at that all the way down the pedigree. So this is called genetic anticipation. It's like the gene is anticipating that it needs to be destroyed, so it starts showing itself earlier on. It's kind of crazy. Kind of like self-destruction. Now, this is a gene disorder, and this is why I'm not sure how it works and stuff like that, how this actually happens. I know in chromosomal disorders this happens also, but this makes more sense to me. Because if you have a messed up chromosome, right, and you're going to make gametes, when that chromosome gets divided and maybe it goes through crossing over, it could get even more messed up, right? Or maybe you have one where it's left out, or you have an additional one. So that one I could see the mechanism for it becoming worse and worse. And Nurse Story talked about this. Do you guys remember? One of her sisters, from her sister having some issues to her children having some issues, she was saying that in her family the symptoms were getting worse. So anyway, that's another example. Now this one, if you guys want to do the extra credit, you really want to listen to me carefully. I posted it up on Google Classroom a couple of days ago. It's an investigation straight out of the investigation manual in your book, okay, or online, and it's about transposable elements. These are also known as jumping genes. Now this is crazy funky too. So we have found examples where there's these little sequences of DNA that are in the chromosome, and they move around within the chromosome. So an enzyme comes and cuts out this piece of DNA, and then it gets reinserted in a different part of the DNA. Now this is a problem if it gets inserted in a gene. You guys follow me on that? If a code, ACTTTTC, whatever it is, all of a sudden is inserted where a gene was, is that going to be a problem? Yeah, it's going to mess up the gene. The protein's not going to be made correctly, if even made at all, okay? And then, but what's crazy too, is they've seen where then it jumps out of a gene. Again, the hypothesis here is it's making diversity. So the example that you'll study has to do with the corn. That's how Dr. McClintock, how she figured out about these transposable factors or units, she calls them. So look up here, you guys. Look at these things. So what are corn kernels? What are they? What are corn kernels? What? Seeds. They're seeds. They're multicellular. Sometimes people get them confused, like maybe they're single-celled, like an egg, right, is single-celled. But it was an egg that was fertilized with pollen, and then it started, doing mitosis and growing and getting larger and larger. Well, check it out. So each of these seeds is made out of a bunch of cells. Some of the cells are expressing the pigment, and some of them aren't. So you get this speckling effect on the corn because of the jumping gene, messing up the gene, sometimes jumping out of the gene. You can see where a lot of it, where there's a lot here, it doesn't seem like it's jumped back in. Okay? So the first part of the extra credit is very basic, and it's a good review on Mendelian genetics, the dominance, and just counting some corn. It's kind of cool. Because every one of those corn kernels is an individual offspring. Okay. The second part in the lab talks more about the jumping gene, and the diagram's a little hard to interpret unless you kind of listen to me carefully or come in. The first time I looked at it, I was like, what? And it really took some thinking, okay? So come in if you're not getting it and you would like this extra credit. I also give you partial credit. If you only want to deal with the first part or part A, whatever it's called, that's fine too. And it's all, everything you need is right there on the pages of your book and just writing it in your journal. Okay, so that's 14. We need to get a little bit of 15 going here. Now, how am I going to assess you on 15? A big vocab quiz, right? Actually, it's not even that big, but a complete vocab quiz. I will quiz you on every word that is on that list in a couple weeks. We're not sure on how time's going to go with all the different things going on in our schedules. We will come back to our garden. We need to get all our data in data tables and stuff like that. So we will come back to that next week. Okay, so you've got to know what the Human Genome Project is. It's a vocab term, and you should just know what it is. Now, you should also know that a lot's been done. For example, have they sequenced all the base pairs of our human DNA? Do they know the sequence of A's, T's, C's, and G's on every one of our chromosomes? Absolutely. I think we're getting close to 15 years. Well, they just know us chemically, pretty well this way. Now, is the project done? No. No, because now we have this big book. I think the analogy was when you guys were watching Power of Genes. Now we have to interpret the book. We have to study the book. We have to figure it out. Now, again, a lot of it has been done. Before I leave this page, though, also this brings up some interesting conversation, right? With ethics and social things, and some people think that it's good to manipulate our genes, some people don't. Oh, come on. Okay, so here's an example, and you can see we got chromosome 21, 22. Here's the famous X chromosome. How many of these do you females have out there? 2. You males only have 1, and then you have the Y. Okay, again, it's not a Y shape. It's just a linear chromosome that's way smaller. Right here, because of course we're most interested in understanding diseases, this shows you where all the genes that can have mutations in them and cause specific disorders. And that's all part of the Human Genome Project. Okay, another topic is functional genomics, where instead of looking at the DNA, they're looking at the functional product. What is a functional product? What's that a model of? Those, yeah, the protein. So now there's been scientists for a while doing this. They're actually studying the protein. So not the code that made the protein. Let's look at the protein. And also, can we affect that protein and maybe not have to do it at the DNA level if there's something wrong with it? Or can we just make the protein ourselves? Okay, so remember proteins, was that a random bunch of amino acid chains? It was very specifically shaped, and you can see that with here. They're trying to model it. So one type of proteins are enzymes, and they help reactions happen. So you see how they perfectly fit to the reactant or the substrates. And again, it looks like because the opposite's attracting, it's going to pull that apart. It can't do it if it's misformed. Okay, real quick, let me go back to this other one. Quick review. What is A? Can you see it? TGTCG8, what is A? A piece of what? DNA. So it's one of the nucleic acids. What's the other nucleic acid? RNA. Why do we know that it's not RNA? Yeah, there's T's instead of U's. Then what is this thing? Cytosine, arginine, glutamine, serine, leucine. What are those? They're the amino acids, right? Now it's missing a messenger RNA because the DNA is transcribed in the messenger RNA. The messenger RNA, right, through translation brings the amino acids, and then we build these polypeptide chains. Okay? So this chapter really pulls in a lot of old stuff because now we're using this stuff practically, and we have to pull from different things you guys have learned. Okay, so this is what we're going to start prepping for really soon. Okay, I'm going to stop at this slide here. So what the deal is we'll read the first bullet. Humans have a long history of practicing selectively breeding, which is manipulating traits in organisms. What am I talking about here? Dog breeding. What'd you guys have for breakfast? So you had wheat, you had peanuts. You're lying. Pancakes are full of wheat. All bread products are made out of wheat. Anyway, these are organisms, you guys. We eat organisms. Whether you think it was alive, I mean, yeah, does pancakes feel like it came from a living thing? Maybe not, but it did. Came from wheat plants. Or there's also maybe some oats in it, another plant. Okay? So anyway, my point is, you guys, I'm almost done, so join me here. My point is, we have been manipulating life for thousands of years. We have been making things breed with each other that would not normally breed. I'm just pointing that out. I'm not saying if that's right or wrong, but I just want to make sure you guys, when you guys are learning more about this biotechnology, and if you're making judgments, if you think it's wrong or right, let's make sure you understand what we've been doing that you seem to be accepting with no problem, or maybe I just don't know that, okay? So, again, just bringing this up. They do. It's fun. I love all this variety of food products. Okay. So genetic engineering. Did you guys have this on your learning guide? Shh, guys, I need you. We're being recorded, and I need to finish up quickly. So, did you guys add genetic engineering to this list? Because I might have missed genetic engineering for this particular class. So add these 3 words to your list. They're on the board. Okay, so genetic engineering, I want to make sure you know exactly what it is. Because since it's in the news and it's being argued about so much, you should know exactly what we're talking about. So when we're talking about doing DNA fingerprints, when we're talking about the Human Genome Project, that is all observing. That's looking at things. Yes, we cut up the DNA, but we got it from some cells from the person that they discarded or we collected from them. I want to make sure you guys understand this. We are not engineering life. You are not manipulating life when you are just looking at the DNA, chopping up pieces of DNA. It's never going to be used in a living organism again. Are you guys okay with that? Okay, hold on. Those 2 things, Human Genome Project, DNA fingerprinting, are not genetic engineering. Now, what is genetic engineering? Biologists prepare recombinant DNA. So we get different pieces of DNA and we put them together. And if we put it in life, right, we put it in cells, and the cell starts using that DNA, making the proteins from the DNA, then we've engineered life. Okay? But again, think about the breeding and things we do also is mixing that DNA. But we're directly mixing the DNA by, collecting the DNA from cells, right? Just extracting the DNA just like you extract it from your cheek cells. And then we can cut the DNA, this is very complex, I'll show you, and get the exact piece of DNA we want. Hold on, we could stay with me. We can insert it into another piece of circular DNA. Now this is a plasmid, and we use the plasmid to move our gene around. We can put it into other cells like this E. coli bacteria. The cell can then transcribe the gene, translate it, and make the protein. Okay, that is making a transgenic, that's doing transformation.
So we could bring the bird back to life.